Dear friends and colleagues, my name is Denny Sarkis.
And my name is David Gardner, and it is our pleasure to welcome you all to the first virtual online Congress on Future Innovations for Embryologists.
We have a fascinating series of lectures for you over the next 6 hours, so please sit back and enjoy. Of course, you will also have access later to view the lectures at your own convenience.
Given the rather large number of registrants for this event, currently we have over 8,000 delegates, and the global nature of this conference, which is spread over many time zones, we encourage you to submit your questions online, and we'll be posting the answers to them in the coming days. And you can type in questions at any time on the right-hand side of your screen.
We'd also like to take this opportunity to thank a few people. Firstly, we'd like to thank our illustrious group of speakers for providing their time. We would also like to thank the sponsors for the amazing exhibition area. We encourage you to visit the exhibition area, which contains plenty of information on new products. This will stay open over the next 2 weeks and is easily accessible through the website. Finally, we would also like to thank the organisers from CME Conferences for all their efforts. And of course, we would also like to thank you for registering and joining us. Thank you for listening, and we look forward to your participation. Welcome to the first session, The Role of the Embryo. Our first speaker is Professor David Gardner, who is Scientific Director of Melbourne IVF and a Distinguished Professor in the School of Biosciences at the University of Melbourne. In 1997, he became the Scientific Director of the Colorado Center for Reproductive Medicine in Denver, where his innovative work on human embryo culture conditions revolutionized how human IVF is performed and in the introduction of blastocyst transfer. He has published over 285 papers and chapters and has edited 15 books.
Our second speaker will be Dr. Dennis Sarkis, who received his undergraduate training at the University of Melbourne here in Australia and received his doctorate of philosophy at Monash University. He has been the laboratory director of IV units in Europe and the USA and has extensive clinical IVF and reproductive research experience. Dr. Sarkis is currently Chief Scientific Officer at Boston IVF and remains an Associate Professor in the Department of Obstetrics and Gynecology and Reproductive Sciences at the Yale University School of Medicine. Our third presenter is Dr. Marcus Meseguer, who received his PhD in Obstetrics and Gynecology and the European Doctor degree in 2002 from the University of Valencia in Spain. He has published over 150 publications, and he is an Associate Professor of the Master in Biotechnology from Valencia University. Our fourth speaker is Angus Tran, who's a world-ranked AI engineer and data scientist. Angus is also a medical doctor who received his training at the University of New South Wales. Angus is the inventor of Ivy, which is an AI model that selects the best embryos for IVF and transfer. He is passionate about applying machine learning to make healthcare better, faster, and more affordable for patients through meaningful collaborations with clinicians. Our final presenter for this session will be Professor Carlos Simon, who is a board-certified and full professor of obstetrics and gynecology at the University of Valencia. He is a senior lecturer at Harvard University in Boston, an adjunct professor in the Department of Obstetrics and Gynecology at Baylor College of Medicine, and the head of the Scientific Advisory Board of iGenomics. He is author of more than 450 publications in international peer-reviewed journals. Hello, I'm David Gardner, and it's my pleasure to be giving the first presentation in this online Congress on future innovations for embryologists. And I'll be talking about how to make a healthy, viable embryo. And here's my disclosure. Now, the pre-implantation period is highly dynamic as the embryo undergoes significant changes in its physiology and metabolism as it develops from the single-cell totipotent fertilized oocyte to the differentiated blastocyst. And during this period, it progresses through the oviduct and to the uterus, which provide very different nutritional and signaling environments for the embryo. Furthermore, though, the The embryo, and this is a cross-section of the human fallopian tube, is in very close proximity to the cells of the mother. So there's readily communication between the mother, the embryo, and the mother with regards to metabolites and signals. And all the time the embryo is moving. Now, if we consider that to the stark contrast of how we grow embryos in vitro. In vitro, here we have two types of incubators. Here we have our standard box incubator, and we would grow our embryos in microliter drops under mineral oil or paraffin oil. And compare that to the new benchtop or time-lapse incubators, and they themselves have redesigned the very dishes that we use, so much so that embryos now reside in microwells, which somewhat helps create a microenvironment around the embryo, but I think we'd all agree it's somewhat different to the environment in vivo. So as a result of working in vitro in the laboratory, we create stresses. So what does stress do to the embryo? Well, you can see stress is affecting your embryos because they grow slower, they can fragment, you get fewer blastocysts, and those blastocysts that do form, you can see the inner cell mass, for example, doesn't develop very well, and you can visualize that quite clearly. However, the embryos can also exhibit altered metabolism, gene expression, and imprinting, which you can't see. The culmination of all of that, though, is reduced viability and hence lower pregnancy rates. So what are the sources of stress on embryos? And if we can identify them, we can help to eliminate them. Serum is a very well characterized supplement, which gratefully we don't use very much anymore, hopefully not at all, which is well known for its adverse effects on embryo development. Poor medium composition, poor formulations can also compromise the embryo significantly, and certainly those media that lack amino acids can be highly detrimental. Ammonium is a problem. It's an artifact of using amino acids in culture. They break down at 37 degrees to produce ammonium, and the embryo themselves can produce ammonia. So one has to bear in mind and design the media very carefully to make sure that toxic levels are not reached in vitro. Oxygen is highly detrimental at atmospheric levels, which is 20% when you're at sea level. It's extremely toxic to both gametes and embryos. I put premature replacement of the embryo in the uterus as a stress. So if you put the cleavage stage embryo into the uterus, that's asynchronous, and it's a different nutritional environment and indeed causes stress on the cleavage stage embryo. So hence the blastocyst transfer. Shear force through pipetting. If one is too rigorous with pipetting, something as simple as that, one can activate stress factors in the embryo and compromise their development. And if there are shifts in temperature or pH, you can alter the homeostasis of the cells, having a dramatic impact on their development. And here I've included a reference that's a nice review covering all of those aspects of stress and how it actually affects the embryo in great detail. So in order to culture a healthy, viable embryo, I would propose that you need to take a more holistic approach to embryo culture. And this is simply due to the fact there's a multitude of parameters in the IVF laboratory that impact the embryo. And it's more than that, they actually interact with each other. And that makes things very complicated very quickly. So this review goes into great detail on many of these stresses and how they can interact. So what I want to do now is provide you a framework on how to analyze an embryo culture system, how to look at your IVF laboratory. So we start with the culture media. And for many years, people would assume that the culture media was the only thing you needed to change in order to get good embryo development. But actually, the culture media is part of a culture system, the system being made of tissue culture ware, how it's tested, the oil overlay, the number of embryos per drop, and the gas phase used. And they are all highly significant. And I include this paper because it's a very nice example of how the gas, atmospheric oxygen in this case, can interact with embryo density. In other words, the number of embryos per drop. Because embryos tend not to like being grown individually, certainly in a microdrop system. The culture system itself resides obviously in the laboratory. And the laboratory has your embryologists, your cells at their training level, the number and type of incubator. From the schematic I showed you before, you could see we have box types, we have bench types incubators, they're changing dramatically. And then, of course, we have to consider air quality, the environment within the laboratory itself, and what kind of purification we have put in place to protect the embryos as they develop. So to run all this effectively, you need quality control, quality assurance. This is essential, and it's really important that we invest in these systems. Particularly, much overlooked is temperature and pH. And putting it simply, the significance of these two parameters cannot be overstated because they have a profound impact on the cells. And really, at this level, we should be able to monitor those with a great deal of accuracy. And this is a very nice review written by pretty much a who's who of embryology. And in that, they cover the significance of temperature and pH. So on top of all of this, now we have the patient, and possibly the patient is the greatest variable in this equation, which will dictate oocyte quality. The patients, of course, they're not all the same. We have different ages, etiologies of different genetics, and of course, different lifestyles, diet, and that plays a huge impact on oocyte quality. And then we add on top of all of this stimulation, which not only impacts the oocyte quality directly, but has the capacity to impact endometrial receptivity. So it's only really by taking a step back and looking at this holistically that you really have the best opportunity to improve the overall performance of your laboratory and therefore grow healthy, viable embryos. Now, it turns out that the embryo is increasingly sensitive to its environment the younger it is. So the fertilized oocyte, the oocyte itself, are highly susceptible to changes in pH because they lack the mechanisms for regulating pH itself. Certainly the fertilized oocyte doesn't have those mechanisms for several hours post-fertilization. The cleavage stages, prior to compaction, are also sensitive to their environment because they're more like unicellular organisms. They don't have an epithelium to regulate their inside and out. So they too are highly susceptible to changes in pH, for example, or medium conditions. But once you've formed an epithelium, the embryo is better able to control its environment. And so much so by the blastocyst, the inner cell mass is really being bathed in the solution that is the blastocele created by the trifecta. So given the fact that the embryo is so sensitive to the early environment, it pays to work in what are known as pediatric isolates or embryo workstations, so you can ensure there is a maintenance of temperature and pH at all times. So what about the types of culture media? Currently, we are faced with two types of choice: sequential media and a monophasic or time-lapse media. So sequential media were developed to mirror the nutrient pool to that in vivo. So the oviduct and uterus provide different environments, and sequential media were designed around those. You have the opportunity to remove all ammonium because you actually change the medium halfway through the culture from phase 1 to a phase 2. They're physiologically based. However, it involves more pipetting and a loss of paracrine factors. And clearly, if you're doing that pipetting outside of a pediatric isolate or workstation, then you're going to have stress with regards to changes in pH and temperature. In a monophasic system, there are no pH or temperature drifts on day 3, when otherwise you would do a changeover. It stays in the incubator. There's less pipetting and hence less stress. There's a cumulative benefit of paracrine factors, so that's all good. However, there's no accounting for nutrient gradients or buildup of metabolites, so it's a balancing act. And I'll come back to that later on. So what about the future of media formulations? Many people have said over recent times that culture medium has been optimized. There is no more future developments in culture media formulations. So this is an area that I've been working in for over 35 years now, and I can honestly say I don't agree with that. There's plenty more that we need to do with regards to media formulations. If we consider the culture systems that we use, currently they're based on salts, amino acids, some vitamins, carbohydrates, albumin, and hyaluronate, the macromolecules. If we compare that to the female tract, you can see there's quite a lot of things that we don't have in, and two in particular are growth factors, cytokines, and antioxidants, all of which are present. And those are the two things I'd like to discuss briefly with you. So when we consider growth factors and cytokines, this is a review that Carlos Simon and my groups put together a few years ago, which really conveys the complexity of the number of ligands and receptors available within the reproductive tract and the embryo itself. In this paper, we were looking at a Luminex assay to look at the different proteins present and with regards to embryo implantation, for example. And what you can see from this, the data that we created, was that there's an abundance of different chemokines, growth factors, and cytokines. And particularly one that stood out at the highest abundance was VEGF, which is, of course, known to play a role in implantation. So, taking on from that, it's present at high levels. We were able to show that the addition of VEGF in a mouse embryo model certainly improved embryo development and implantation. But that's just one factor. Here's another, GM-CSF. Now, although we only found this at low levels in the human uterus, there is a significant amount of interest in this. And this is a randomized clinical study done by Soren Zeeb and colleagues. And in it, they found that there was a protective effect of GM-CSF on culture-induced embryo stress. So when the culture medium itself had low levels of albumin, GM-CSF was able to protect the embryos from stress. So it could be that some of these growth factors are actually acting as survival factors. And indeed, Kate Hardy showed this to be true with the IGFs many years ago. This is a very nice paper that was published by Aaron Shu and colleagues. And what they did was actually look at a group of growth factors. Rather than look at them individually, they looked at a group. And it was quite a large group of brain-derived neurotrophic factor, colony-stimulating factor, epidermal growth factor, GM-CSF, insulin-like growth factor 1, glial, cell line-derived, etc. And they looked in a serum-free medium, and what they showed for normally fertilized embryos on day 3 that was surplus, they got an increase in embryo development and embryo quality. So this is very encouraging that a group of factors would be beneficial. However, what we need to do, though, is consider appropriate epigenetic analysis and long-term follow-up of those individuals. And we should do that in a mouse model first, because clearly with that number of growth factors, some are acting as survival factors, some are acting as mitogens. They're all signaling, and some will be there to drive the embryo, some will be there to regulate the embryo. So I think it's very important that before we dive in, we need to ensure the epigenetic state and long-term health of those offspring have been carefully assessed. And now let's turn to antioxidants. This is something that we've been working on for a number of years. So I alluded to the fact that oxygen is a stress. In vivo, fertilization embryo development occurs not at 20%, not at atmospheric, but at a much reduced concentration, typically around 2 to 8%. And it's been shown in pretty much every mammalian species today that culture at 5% oxygen increases embryo development and increases viability. And the very last study I showed there from Marius Mencius shows that culture at 5% oxygen improved blastocyst quality and development and led to an increase in live birth rates clinically. The negative effects of atmospheric oxygen are numerous. In fact, it's hard to find anything in the literature that has so many detrimental effects on embryos. So I've listed a few here: gene expression, epigenetics, the proteome, metabolism, cellular homeostasis, cleavage, blastocyst development, and viability. So it would seem logical to avoid at all costs atmospheric oxygen. And here's the other catch with oxygen. 20% oxygen increases the vulnerability of gametes and embryos to other stresses. So by exposing embryos or gametes to 20% oxygen, you make it more susceptible if there is a second stress present. And this has been documented time and time again. I've showed the example initially of individual culture. There's data that shows that actually the embryo can no longer regulate its ammonium transamination in the presence of 20% oxygen. It completely changes the physiology of the embryo. So we need to avoid it and reduce its effects. However, atmospheric oxygen, even if we grow our embryos in 5%, we still expose embryos to atmospheric oxygen during a collection, preparation, ICSI, FERT checks, grading, biopsy. We should not grow embryos in 20% oxygen. As a result of which we get reactive oxygen species formed, and it's known to be detrimental to sperm and in cryopreservation. And I should note, even at 5% oxygen, oxidative metabolism can create reactive oxygen species. So even at 5% oxygen, we still get oxidative damage. So what happens with the oxidative damage? Well, we can get poor embryo quality, high levels of peroxide and fragmentation, cell membrane and DNA damage. impaired oocyte reproductive ability, and sperm is compromised. So it clearly has an adverse effect on gametes and embryos. So what can we do? Well, antioxidants have been added to culture medium for a number of years. Historically, we started back in 1991 when NODA was adding specific antioxidants, but they've been added individually. It's been shown they can prevent oxidative damage to gametes and embryos, and there's a numerous numbers of studies that have shown they improve embryo development and competence. Our question was, what happens, however, if you look at antioxidants as a functional group as opposed to being individuals? And this is the first paper we published looking at a group of antioxidants on mouse embryo development. We screened several antioxidants and came up with three that we were extremely interested in. That's carnitine, alpha-lipoic acid, and N-acetylcysteine. They all have different roles to play. Alpha-lipoic acid, otherwise known as the antioxidant of antioxidants, has the capacity to reduce other antioxidants once they've been oxidized. And so we built upon our work on lipoic acid and included these other antioxidants. So what we wanted to do is we worked out the optimal concentration for each antioxidant, and then we wanted to know what happens when we use them together as opposed to individually. And what we found was that the triple antioxidants had a highly beneficial effect of embryos grown at 20% oxygen. And this is cell number of mouse blastocysts grown in 20% oxygen. And we grew embryos in groups, and we got a 15% increase here in total cell number. And when you grew embryos individually, we got an even larger relative increase of 34%. But you can see that by growing embryos individually at 20% oxygen, that is indeed such a stress that we got a significant reduction in the cell number of this group. So what we took from this initial study was that the triple antioxidants have a greater beneficial effect than the individual ones, and that, there we go, and the triple effects are greater on individual embryo cultures. We then repeated this experiment and looked again at 5% oxygen. Again, we used embryos in groups and embryos grown individually. And what you can see is that whether they were grown in groups or individually, there was a beneficial effects of antioxidants even at 5% oxygen. And this is because, of course, reactive oxygen is formed even at 5% through metabolism. But what we saw was that the relative increase was the same between the two. When we did embryo transfers, what we saw was our control group here, and this is our antioxidant group. Now, we've been doing a lot of embryo transfers in our animal research for many years, but it was very rare that we saw such a dramatic improvement of something in culture that translated to fetal development. So much so that we saw a 14% increase, significant increase in fetal weights, in placental weights, and in crown rump length. We saw an elevation, a difference of 10% in implantation, but it didn't reach statistical significance. So that was very encouraging. So we then went on to look at the effects of antioxidants in IVF. The previous study had been used fertilized eggs in vivo. So we wanted to expand the studies and look at IVF. And in summary, what we found was that this is the IVF or culture. The control was no antioxidants. The A3 is 3 antioxidants. And this is our control. This is the control. So the control, no antioxidants in the IVF, only in the culture, and we saw a beneficial effect. Vice versa, if we added the antioxidants just to the IVF, but not to the embryo culture, we got a beneficial effect. But you can clearly see that when the antioxidants were present in both the IVF medium and the culture medium, we got an even greater effect. And then more recently, we looked at the effects of antioxidants and their role in cryopreservation. Specifically, did they have a benefit when added to the cryoprotectant solutions during vitrification? And again, we saw, indeed, they did, because vitrification is associated with a generation of reactive oxygen. And what we saw in this case was, again, significant increase in fetal weights for embryos that had been grown at 20% oxygen and then vitrified with or without antioxidants. And again, increased crown length, limb morphology, and fetal age. When we repeated this for embryos cultured at 5% oxygen, We still saw an increase in crown rump length, embryo development, but we didn't see an increase in fetal weight, although we did see a 12 milligram increase overall. It wasn't significant. But we do see an increase in overall fetal development. So to summarize these animal studies, we showed that a combination of carnitine, lipoic acid, and acetylcysteine, either in the IVF or culture medium, improved embryo development at both 5 and 20% oxygen. The effects are greatest when you have them throughout the system. So have IVF antioxidants present for IVF, have antioxidants present for embryo culture. We also showed that presence of antioxidants in the handling medium helped to reduce levels of intracellular peroxide and actually maintain levels of glutathione. So that was a very positive result as well. And as a result, fetal development was increased in the combination of the 3 antioxidants at both oxygen concentrations. And finally, we've shown that antioxidants protect embryos during cryopreservation. So what about clinical studies? This is a paper that came out this month, whereby in a collaboration with Kuramoto and Yoshida in Japan, we were able to look at sibling oocyte splits and the effects of the antioxidants on human embryo development. The embryos were then vitrified and subsequently transferred, so we got pregnancy data as well. And here we see the embryos that were cultured in our standard culture medium, for patients under 35 and 35 and above. And this is the implantation and fetal ongoing pregnancy rate. And what you can see is that in the control group, there's an age-related decline in both implantation and ongoing pregnancy rate, which is typical. However, when we looked at this group over here, which had the 3 antioxidants that I've discussed previously, what we saw was that we saw a maintenance of implantation and pregnancy rates, which was really exciting. So it looks like that the presence of antioxidants in a human culture system can somewhat alleviate the age-associated decline in embryo quality and viability. And this is consistent with the model that as the patient ages, as the gametes get older, they're more susceptible to oxidative stress, which accumulates through their lifetime. So currently, we're doing a prospective randomized trial at Melbourne IVF to expand this study and to look at these age-related differences in responses to these antioxidants. So my concluding thoughts on how you grow embryos. I think it's very important that you take a holistic look at your laboratory. Embryo is so much more than culture medium. If you buy a culture medium and expect to get the same results as in a laboratory, you may be disappointed if you haven't invested in the same degree of technology, air purification, etc., etc. It's essential that you invest resources into comprehensive quality control, quality assurance. Some people may not think this is the most exciting component of what we do. I can assure you it's one of the most essential things that we do. And quality management systems give you the feedback that you need to make changes quickly if you do indeed identify a problem. I think the efficacy of sequential and monophasic systems is dependent upon how your laboratory is set up and your patient volume. If you're a boutique clinic working with isolates, then sequential medium may work very well for you. But if you're a very large program with high volume, then maybe the monophasic is your way to reduce the overall stress. Remember, it's a balance of all the stresses. Certainly, I think that growth factors will become part of our culture environment in the future, but it would be great to see some detailed analysis on the epigenetics and long-term follow-up of the embryos created through exposure to growth factors and cytokines. In a mouse model, I think it's really clear that antioxidants as a combination improve IVF, culture, embryo transfer, and vitrification, which is really exciting. And as I alluded to, antioxidants may indeed help in alleviating the age-related decline in human embryo development and viability, which is a very exciting prospect indeed. I think it's important to reconsider that culture has not been optimized. We are all working very hard to further advance the environments that we hold our gametes and embryos. So with that, it's a great pleasure to always acknowledge the team that I work with at the university and at my IVF clinic, particularly to Mai at the university, who was a real driver in the antioxidant studies, to John and my team in the embryo lab, and to the docs at Melbourne IVF, and to my colleagues Jeremy, Denny and Carlos, etc., with whom I've worked for many, many years. One couldn't really talk about embryo culture without referring to Michelle Lane, who sadly passed away a few months ago. She was just 49. Mitch was my first graduate student. She was my postdoc. She was my colleague. She was my friend. And she'll be greatly missed by many. I think it's always important to remember the massive contribution she made to our field. Now, I've thrown a lot of information at you, and here I've left some key references for you to consider. You'll notice that the top three all have Michelle's name on, and the very last one was we wrote the chapter together for Peter's book 2019 on embryo culture. I didn't talk about the components of embryo culture media, the effects of pyruvate and lactate concentration, which amino acids you should add and when. I would have taken your whole day up to do just that. However, the chapter I've highlighted, the 2017 reference, is probably the most comprehensive analysis of human embryo culture medium I've ever read. And I'd encourage you, if that's of interest to you, to have a look at that chapter. So with that, I thank you very much for your attention.
Welcome to the first online embryology conference, and today I will be presenting some new data on non-invasive embryo selection by metabolic imaging. This project is done in collaboration with a group that we collaborate with at Harvard University, led by Dan Needleman. So we're going to hear a lot today about different technologies for picking the best embryo. And of course, the one that we use mostly is morphology, but we'll also hear about morphokinetics and hopefully some other non-invasive techniques. But what don't we see when we're looking at an embryo, for example, which is what we're doing? But I'll give you one example of when you're looking at people, for example, and what you don't see. So let's take Winston Churchill, Roger Bannister, and Jimmy Fix. You can see that they all had various characteristics, and obviously the two on the right-hand side were very fit. But who lived the longest? And of course, this is a fixed slide, so you could pick that Winston Churchill actually lived the longest, even though he actually lived badly, you could say. So there are a lot of things that by just looking at people, we don't really see. And our cell of choice, or what we want to look at, is the embryo. The pre-implantation embryo is actually quite amazing in the way it metabolizes. It actually has a very intrinsic pathway that it actually chooses during the pre-implantation stage. And of course, we're all aware that during the cleavage stages, it has a preferential use of pyruvate and lactate using the Krebs cycle or the TCA cycle. But during the 8-cell or compaction stage, nearly all mammalian embryos will switch to a preferential use of glucose and abandon the preferential use of pyruvate and lactate. So this is a very interesting switch that actually we can use, and we have used historically, to test and actually to define culture media. And of course, key to this event is the mitochondria, and I'll get back to the mitochondria in a few minutes. So the problem is, how do we measure that? We can't just look at an embryo and say, oh, it has a good metabolism or a bad metabolism. So many people have worked for many years to actually try to understand the uptake, the production, and of metabolites that are key to mitochondrial metabolism and the embryo metabolism. We do, though, have some interesting technologies that are semi-non-invasive that may allow us in the future to use this information of change in metabolism to actually help us to pick which is the best embryo. And I'm going to talk about some of these technologies today. So the metabolism of an embryo is very interesting. And the key hypothesis that many people have been working on is that the assessment of specific metabolic pathways, their relative activities and their regulation is related to embryo viability. And of course, we can look at this metabolism by just looking at key factors such as glucose, lactate, or pyruvate, or trying to measure cell functions like the TCA cycle or glycolysis. And it's been known now for over 40 years that glucose metabolism can be linked with the viability of embryos. This was first shown in France by Jean-Paul Renard in bovine embryos with these Menezo. So it's been a long time that we've known this actually works, but it's been very hard to show. But a lot of people, including people that are speaking today, David Gardner and his mentor Henry Leese, have worked for a long time to actually show that glucose consumption or glucose uptake is actually a fantastic marker for embryo viability. And they've shown this in multiple species, and they've shown it, of course, in mouse. But several years ago, David Gardner and Michelle Lane showed that glucose consumption of single post-compaction human embryos was predictive of not only live birth outcome, but interestingly also live sex, of embryo sex. So what is interesting is that the higher the glucose uptake, the greater chance that an embryo has of being viable. So this is really proof of principle that metabolism is a key indicator of embryo viability. But the trick is, how do we measure this? What is an easier way to measure it than some of the complicated techniques that have been available for many years? What about using different types of semi-non-invasive technologies? And one of the ones I'm going to talk about today is fluorescence lifetime imaging microscopy, one that we've been working on for several years now with some collaborators. But there are many optics and photonic systems that could be used to actually get information from embryos. And I'm going to touch briefly on some of these, but mostly I will talk today about a laser technology, the fluorescence lifetime imaging microscopy. And what does this technology allow us to do? So non-invasive fluorescence lifetime imaging microscopy, or FLIM, allows us to measure embryo metabolism. It's been used for many years to measure cellular features that are actually been interested for research. And of course, it allows metabolic activity to be measured by looking non-invasively at the function of mitochondria. What it allows, it's got a long wavelength, it has a low power light, and of most interest, no reagents are needed to add to the embryo or the cell of interest. So what can we look at? And the two factors that we're most interested in are NADH and FAD, which are both important intermediates of the electron transport chain. Both NADH and FAD are essential to cellular respiration and ATP production. And what it may allow us is to look at this change in metabolism that the embryo undergoes and to look at the quality of that metabolism. One of the key factors, though, is that both NADH and FAD both autofluoresce. So there are no need to add markers to the embryo. So all this is non-invasive, and you don't need to actually add anything to the culture media to test the metabolism of the embryos. So how can we do this? So fluorescence lifetime imaging microscopy looks at both these uses excitement of fluorophores to take time to de-excite. So it investigates the time the photons are actually excited and how long those photons take to actually de-excite. So the FLIM creates an arrival time for histograms of fluorescence photons, and what we're interested in are both NADH and FAD. So we get a lot of information about how these photons are actually behaving within an embryo and within the mitochondria of an embryo. So we published several years ago, or a few years ago now, this paper on combined non-invasive metabolic and spindle imaging as a potential tool for embryo and oocyte assessment. So the data I will show you initially are all taken with the mouse. So this is the type of image we get. This is a one-cell embryo and it's an NADH image. You can see here the clarity of the image, and you can use the same process for FAD. So what happens is you generate a FLIM timeline, and you can look at the histogram of the photon counts of NADH. This gives you quite a lot of information. It gives you measurements and parameters for each embryo for both NADH and FAD. It gives you the brightness. It allows you to work out the fractions bound to the enzymes, and it tells you about the lifetimes of the fluorescence. So all in all, both NADH and FAD, you get 8 parameters that are all sensitive to the metabolic state. You can also work out the redox potential of the embryos too. So this is an example of mouse embryos. On the left is NADH and on the right FAD, and you can see the differences in the brightness only of the embryos as they go from the one cell stage to the blastocyst stage. So you can see here on the left, the embryos are quite bright in the earlier stages, and then there's a switch as the embryos on the left actually get less bright, and on the right-hand side they become more bright. So you can actually plot this, and you can see here that the embryos' NADH is in the blue rings, and you can see it increasing, and then as they get to the Morley stage, they switch to a different metabolism, and you can see the NADH fluorescence actually going down. In the green, you can see as the embryos grow from the 2 to the 4 cell stage, they're still quite low, but as they increase FAD usage, they actually go up post-blastocyst stage. So the type of patterns we observe give us a lot of information about how the embryo is behaving metabolically. Of course, this has been known for several years, and what we have known is that this mimics exactly what Henry Leese and colleagues published many years ago in the mouse about glucose metabolism going up post-compaction, and actually pyruvate and lactate metabolism going down post-compaction and being used more proficiently at the earlier stages. So what we can actually do, we can actually use this to look at metabolic timeliness to distinguish embryo quality. So we can look to see how embryos at the 2-cell stage, 4-cell stage, 8-cell stage, and the blastocyst stage are behaving, not just in terms of their cleavage rate, but actually in their metabolic rates and their metabolic behavior. So the other thing we can do is actually not just looking at the metabolism, but we can also use this type of microscopy to look at spindle imaging. So you can see here a 1-cell embryo going to a 2-cell, so you can see the spindle there appearing and disappearing. So this type of information, in addition to the metabolic system, could actually maybe give us information about if aberrant cleavage is occurring with the spindle not behaving, especially up to the 8-cell stage. So it may also allow us to non-invasively image aneuploidy in certain embryos, or the risk of aneuploidy in certain embryos. If you look here, these are 2-cell embryos, mouse embryos, and you can see the clarity of the image you can get, and you can actually see that you can segment the NADH, for example, and look to see if it's mitochondrial or cytoplasmic. So we can even get more information by looking at the areas of mitochondrial metabolism. So this, in addition to the parameters we get from the FLIM, gives us even more information about how an embryo is behaving in terms of its metabolic state. Of course, we can also investigate the effects of chemical and physical factors on mammalian embryo culture. And I'll just show you one example of this in the next slide. So what happens when eggs or embryos are actually put under a state of hypoxia? So this is actually a question that many people are asking these days, because we're starting to ask ourselves if 5% oxygen that we use in routine human embryo culture is efficient, or can we even go lower? So this series of experiments wanted to let us know how embryos cope with anoxia. So here we actually could live image embryos, and you can see here these are embryos exposed to 0% oxygen just to see how embryos of different stages cope with the anoxic environment. This is a 1-cell embryo, and you can see here the hot patches that are live image, and as the oxygen is restored, you can see this goes away. So you can see the embryo is able to cope quite well with this, but it actually does stress. It does stress the mitochondria. But using this type of technology, we can actually use it to see how different stages react to different changes in our case, oxygen, but also maybe other metabolites that we can see in the future. So what we found here was that embryos of early stages really reacted badly to anoxic environment, while compacted and blastocyst mouse embryos actually were able to cope with the anoxic environment quite sufficiently, which is interesting given what we know about embryo development and embryo pathways in relation to oxygen environment. What about other systems that we can use the FLIM technology to look at? So this is a series of experiments we did with Emery Sally's lab at Yale University, who had a knockout model of a mitochondria that detects that looks at mitochondrial dysfunction. And what we did show here is that the knockout model, which was a protein called caseinoleutic peptidase P, and what you can show here is that If you look at mitochondrial DNA measurements, you can really not see much of a difference between the wild type and the knockout model, whereas when you look at actually various metabolic features, you can really distinguish differences between the knockout and the wild type model. This is interesting for a knockout model where it's a very distinct disturbance in the mitochondria, but what about young and old mice? So young and old mouse oocytes are it's very problematic or very controversial to show differences in their quality. Here again, mitochondrial DNA measurements showed very little difference between young and old mouse oocytes, but actually when we looked at metabolic imaging and quantitative parameters of metabolism, we could distinctly show which were the old mice oocytes and which were the young mouse oocytes. So it's a very powerful technique in being able to distinguish between these two types of oocytes. Of course, if you're going to use a technique like this, and possibly use it in the human, you want to use it to make sure that it's safe. And these are just some simple experiments we did where we illuminated mouse embryos during the whole development stage to see if they were actually affected. And this shows that hopefully it's relatively safe, because we showed that both the number of mouse pups and their mean pup weight were not significantly different to non-illuminated controls. What about human work? So we're currently in the process of looking at the characterization of discarded human blastocysts, and we've looked at over 200 biopsied PGTA embryos, 100 non-biopsied blastocysts, and 100 discarded blastocysts. And what we're able to do is, again, get image of these blastocysts of both the inner cell mass and trophectoderm, and we can look at both the NADH on the left and FAD parameters on the right-hand side. So what type of information are we getting? I won't show you all the information, but what we do get is we can also image the inner cell mass and trophectoderm. We can compartmentalize the areas and look at inner cell mass fluorescence and trophectoderm fluorescence to distinguish if there are differences between certain parts of the embryo also as it develops. So this type of information has already given us an analysis of the metabolic detection. And what we have seen is that there are differences between aneuploid and euploid embryos, blastocysts. There are distinct differences in the metabolic signatures between the inner cell mass and trophectoderm of discarded human blastocysts. So these are all the parameters we've measured are significantly different between the inner cell mass and trophectoderm, which is very interesting. And we see a large change in NADH and FAD between day 5 blastocysts and day 6 blastocysts. So already this FLIM imaging technology is providing us very interesting information about human blastocysts, and we hope to share that with you in the future. The other thing it can do, in addition to the possibility of using our understanding of metabolic imaging, we may be able to get information about viability, but hopefully in the future it may assist us in real time to help develop better media for embryos and understand the intricate needs of embryos at different stages a bit better, especially related to their metabolism. FLIM is not the only technology that's around that can allow us to do these things, and I'll just point you to some other interesting technologies that allow us to look at both metabolism and other factors in embryos. And all of these are non-invasive techniques that are being developed that are linked in some way to some sort of microscopic technique. One of the most interesting is being performed by Jeremy Thompson's group in Australia, and this is a publication using a technology called hyperspectral microscopy. This is a fascinating technology that not only looks at maybe 2 factors, such as we're doing with NADH and FAD, but it actually can look at a greater range of autofluorescent proteins or factors and measure those in embryos. So you can see here that they're showing more than 2 factors that actually are able to be picked up autofluorescently that may be related to, in this concentration, looking at differences in embryos that were cultured in 7 versus 20% oxygen. So this is also a very powerful technique that hopefully will be developed in the future. There are even other techniques, and in the future we may be able to even do nuclear magnetic resonance on individual embryos, and this is a technique being developed in Switzerland by a group that are able to do individual embryos and actually get NMR data on those individual embryos, which could be quite fascinating and give us a lot of information about embryo development. Other systems have been able to develop live cell imaging of the chromosome dynamics. So again, this is similar to what we've been doing with the spindles, but actually looking at more intricate dynamics of cleavage. So this actually may be a secondary way of non-invasively detecting aneuploidy, and we'll hear more about that later today. And of course, there are even more complicated techniques, and this is a fascinating paper looking at bovine embryos, where they've used a technology that looks at micron scale movements within an embryo and actually is able to measure those and correlate those to viability. So all these technologies are being developed and hopefully will allow us to non-invasively select better embryos. And of course, for many years now, we've had types of Raman microscopy technologies that hopefully will allow us not just to look at embryos, but also the gametes. And there have been a number of technologies involved with Raman microscopy that have been published over the years that hopefully will allow us to use this type of spectroscopy to image embryos and possibly select them in terms of viability. So I think non-invasive microscopy and, of course, metabolic imaging is an area that actually, as a discovery platform, is really growing in these years, and hopefully we'll be able to use these types of technologies in the near future to help us assess embryos non-invasively and give us greater information about the viability of embryos. And finally, I'd just like to go through, so what will the future look like? What type of systems will we have? So maybe we're going to have types of microfluidic chips, which we're going to hear about today, where we can actually adjust their culture conditions. So embryos will be in individual wells grown in a microfluidic chip, but within these wells, we'll be able to do various analysis, not only non-invasive microscopic imaging, but also analysis of the media, and to be able to swap the media as the embryos grow to maximize the potential of each embryo and to understand the potential of each embryo as before we transfer it or before we freeze it. All this will hopefully improve the chances of patients to get pregnant and our ability to help infertility patients. So leaving you with that, I'd like to thank you all for listening, and to also thank my collaborators from Harvard University, especially Marta Venturis and Jamin Shah, who have done a lot of the human work, Boston IVF, the embryologists and staff for their assistance, Emily Seidler, who did the mouse work, Tim Chanches, who has helped us over the years, and of course, Emery Selly's lab, and for a long time collaboration with David Gardner. Thank you, and I'm happy to answer any questions. Thank you.
Hello, everyone, and welcome to the first online Congress on Future Innovations for Embryology. I would like to thank Dr. Denise Sakas and also Dr. David Garner for inviting me to give this lecture. I'm Dr. Marcos Meseguer. I'm an embryologist. I am also a scientist and working like assistant professor also in biotechnology in IVA Valencia in Spain. And I'm going to give a lecture about the time-lapse technology and what is the current situation now. First of all, I would like to talk a little bit about the technology by itself and how it has been evolving the last years. So the first thing that I would like to mention is the evolution that this technology has seen in the last years. We started with single point morphological observation on day 3. And that was what we did in the late 80s and 90s. And actually, we're still doing this type of observations. But it was around the late 90s when we started to perform continuous embryo monitoring. And we started also to do blastocyst culture. But it was around 2011 when this technology became popular, let's say, when we were able to analyze several embryos at the same time by using this technology. Putting together all this technology together, we were able to take a look 2,000 of embryos at the same time, and we were able to elaborate a manual algorithm that helped us to select the best embryo for transfer. In the last couple of years, it appears automated platforms that were able to perform the analysis automatically. And then was a big change in the concept of time-lapse because it was the first introduction of the automation in the embryology field. So this is the agenda of today. So we are going to talk about the dynamic changes in morphology, how they are time dependent, and how they categorize the embryos. We're going to take a look to the concept of precision embryology and how we can use it, the identification of atypical phenotypes that are important for embryo selection, how the blastocysts re-expand after warming. Also, we will go through automation and the introduction of the automatic annotations of morphogenetics and morphology. We also take a look to algorithms for embryo selection and how they can be combined by morphology and morphogenetics in the non-KIT score. And also we will go to time-lapse comparison versus standard incubator, and in particular the comparison of different time-lapse systems. So first of all, we will go through what we have learned about time-lapse technology in the last years. First of all, we can see how the dynamic changes in morphology are time dependent, and the categorization of the embryo is important because the changes in morphology may make us to take a decision or select or define an embryo like a good or bad morphology. So it's a good example of an eye to look in the E3 embryo. The rich blastocysts are different aspects of morphology depending on the time, with some blastulation, with some vacuoles, etc., that may help us to decide which embryo we want to select. Also, here you have a couple of videos of two embryos that are growing in parallel with relatively good morphology on day 3, with good number of cells, blastomeres, good symmetry. But when they became to reach blastocyst, one of them, the one in the left, start to generate vacuoles and suddenly start to degenerate. Meanwhile, the other, even that is growing, has some vacuoles, but is expanding quite well and is presenting some contraction. So two very nice, good-looking embryos on the 3 that are following different evolution time-dependent. So this is why the dynamic changes in morphology are important and how this time-dependent categorization may be relevant for selection. So when we do embryo selection and when we apply this type of concept, we can do what we have named like precision embryology. Precision embryology means that we can take a look very accurately to many aspects of embryo development. I mean, we can go through, for example, the time of pronuclear fading. We can take a look to the time of the pronuclear formation. We can go through all these aspects of embryo development, and we can link those with the implantation potential. And everything can be done very accurately. I mean, we can take a look to all these parameters very accurately and with very specific detail. So using this technology of the embryo viewer, we can go through, study things like the dynamic changes in the morphology that are related with the outcome. One of them, for example, is the blastocyst expansion. We can use the technology of time-lapse to measure which was the final diameter of the blastocyst prior to transfer. So this is a very objective parameter that we know from this study that was done by one of my PhD students, Lorena Bori, that presented in ASRM a couple of years ago. She was able to demonstrate a very nice link between the diameter of the blastocyst and the implantation potential. Also, she measured the area of the inner cell mass and find a link between the area in micro square meters and the outcome. I mean, she was able to find that there was a area above 2,100, 2,200 square meters, micro square meters, that were linked with higher implantation potential. And also, for example, how the diameter was growing, and she was able to define categories with different diameters that were linked with higher implantation potential. So this is a very good example of how we can use time-lapse to measure parameters of the blastocyst very objectively, and that are very nicely linked with the outcome without using the subjectivity of the embryologist. Also, in this particular study, she was able to describe the duration of the cell cycle of arthrophoto themselves. First of all, just taking a look to the one blastomere and do a follow-up to the next cleavage and the next one. So she was able to do it in every blastocyst and was able to quantify what was the duration of the cell cycle of arthrophoto cell. The trophectoderm cell cycle was in average shorter than the other cell cycle that has been described before, the first, the second, or the third. So let's say the longer is the time of development, the shorter is the cell cycle. But additionally, she was able to describe that when the cell cycle is below 8.66 hours, the implantation potential of those blastocysts was significantly higher. So it's the first time that the duration of the cell cycle of trophectoderm cell is linked with the implantation potential. So together with that also, during these years, we have been able to identify atypical phenotypes. This is data that we presented a couple of years ago in ESRE. Previously, we published a paper in Human Reproduction in which described the presence of this blastocyst collapse. Blastocyst collapse means that the whole surface, or at least 50% of the surface of the trophectoderm, lose contact with the zona pellucida. It's happening around 20% of the blastocysts, and we have seen that this incidence is related with the implantation potential. We have seen that embryos that present a collapse, in average, implant 10% less. But what is even more important, what we have seen, is that embryos with better morphology, but with the best morphology, but with the presence of a blastocyst collapse, implant quite much less than embryos with worse morphology, but without the collapse. So this is a good example of the interference between an atypical phenotype and the standard morphology. We may select a blastocyst, which is very nice, good looking, but that presents an atypical phenotype that we know which is linked with lower implantation potential. This is the example of why it's so important to identify this phenomenon. The other thing that is important also to see is the behavior of the blastocyst after warming. So here in this video, you see how this is my colleague from Valencia is measuring the initial area of the zona pellucida of the blastocyst, the initial thickness of zona pellucida, and also what is the final area of the blastocyst after 4 hours of culture, and what is the final thickness of the zona pellucida. So we can combine these two parameters and link that to the implantation potential of the blastocyst. So this is an example of how you can do this type of application, or how you can link with the outcome. So let's to the results of this study. If you take a look at the data, you take a look to the data that she presented, and here you have a video on how the blastocysts behave after warming. When we do the warming, the blastocysts collapse, and then we see a re-expansion. So when you take a look to this behavior, to this re-expansion, we have realized that depending on what was the initial area of the blastocyst, what was the final area of the blastocyst, what was the initial thickness of the zona pellucida, and the final one, we have seen that this is very nicely linked with the outcome. For example, the embryos that normally implant trend to start the process of re-expansion 15 hours before the embryos that do not use to implant or have a live birth. Additionally, we have seen that the initial area are the initial thickness of the zona pellucida, and the final thickness and the final area is linked with the outcome. I mean, the bigger is the final area of the blastocyst, the bigger also is the outcome of the blastocyst. And also the bigger initial area, the bigger the outcome. The same with the thickness. The shorter the thickness of the zona pellucida, the bigger the outcome. So what we have realized, and we demonstrate in this publication that we did in fertility and sterility, again, this is the PhD work of Ayla Coelho. What we were able to demonstrate was that an embryo that has a good behavior after warming and is presenting a good re-expansion and is performing the right timing during these 4 hours, this behavior is more relevant for the outcome of that blastocyst than the morphology. I mean, we demonstrate that it's more important to observe the blastocyst after warming than just take a look to the morphology. And even more, that the morphology of the blastocyst is less relevant than the behavior of the blastocyst after warming. So this is a good example of how the evaluation of the embryo by time lapse is sometimes more relevant than the concept of morphology. So the other topic that I would like to underline about time lapse is automation. When I'm talking about automation, I would like to spend my time in one study that we're just finishing now with more than 20,000 embryos using two types of time-lapse systems, Embryoscope and Embryoscope Plus, with single stem media, and taking a look to around 4,000 embryos evaluated by KidScore used in day 5 in the version 2 and close to 15 in the version 3. So here you have a video of how the KidScore works. I mean, KidScore is a commercial software which is present in the embryo viewer of the embryoscope. It has two versions. The last one that we were using is version 3. This version 3 is using some of the parameters of time lapse, like pronuclear fading, time to cleavage to 2, 3, 4, 5, the time of blastulation, and also is doing automatically the classification of the trophectoderm and inner cell mass. And also is considering direct cleavage and also embryo development speed. So everything is performed automatically. I mean, everything, all the timings, even the morphology of the inner cell mass and trophectoderm. So we end up with a classification that is using all these timings, and we have a value, we have a score that we may potentially use for selection using these values. So we have like a ranking from the top to the lower value, and sometimes when we have clusters with very good morphology, it can be very useful. So it's like the annotations are performed automatically. Even when the annotations are done automatically, what we have is a KidScore, which is a score that is helping us to select the best embryo for transplant. So which are the results of this technology? Well, we are comparing versions 2 and version 3. And we first of all take a look between the relationship between the morphological grade that we use with ASEVIR, the Spanish Society of Embryology, criteria, and the KidScore day 5. The first thing that we saw is that the embryos with best morphology with ASEVIR are the embryos with higher KidScore, which makes sense because KidScore is using morphology like a value for the score, like itself. But the difference is that this classification was performed by the embryologists manually, and when we compare with KidScore that is fully automatic, is very nicely linked. So let's say that the system is able to classify the embryos like the embryologist in a very similar way. When we take a look to the implantation rate in relation with the value of the KidScore, we split the KidScore in 4 categories: less than 2.5, 2.5, 5, 5.5, 7.5, and more than 7.5. And we take a look to the implantation rate. This is kid embryos with no implantation data, so it's not the full database. This is for version 2 and version 3. This is groups with similar interval length. You see that in the 2 groups with higher KidScore, we saw higher implantation potential. From the version 2, and I would say that from the version 3, even more accurately and more linear. When we split this by quartiles instead of groups of similar interval, then we see more clearly the difference, but especially in the version 3, from 41% to 67% implantation rate per blastocyst. So taking a look to the relationship between this live birth rate and KidScore, again, with the version 2 and version 3, using quartiles, you see a very nice relationship that is here represented graphically between the values of KidScore, the lower, less than 5, to the bigger, to the higher, with more than 7.5, from 20% to 48%. So we were able to find a very nice link between this KidScore and the live birth rate, which is a score that is doing automatically the classification of the embryos. We took a look also to the chromosomal content in our PGT program and compared with the KidScore. This is the version 2 and version 3 with close to 2,000 embryos in the version 3 and ploidy. The euploid embryos in average presented higher KidScore value than the embryos aneuploid, and this was observed for the version 2 and version 3. And if we take a look to the euploid rate in version 2 and version 3 of KidScore, And the key score values classified or organized by quartiles and euploidy rate, you see a nice linear link between those values and the chance to be euploid. More relevant, I would say, in version 3. So I would say that version 3 has improved the ability to distinguish between embryos with different euploidy rate by using this classification. I mean, embryos with a KIT score below 3.4 has a chance to be euploid to 34%, and embryos with a KIT score above 6.5 has a chance to be euploid or close to 60%. If we take a look to the relationship between implantation rate and KIT score day 5 in good quality embryos, so this is only good quality embryos, only A or B following a severe criteria from Spanish Society of Embryology, we observe that embryos that implanted in average present higher KIT score values than embryos that did not implant, from 6.37 to 6.79. That was significant. Taking a look again to the proportion of euploids and aneuploids, and this categorization, again, we see this difference that were significantly different. So we have seen that there is a system that is performing the automatic annotations of the embryos, very well performed, and also is making you a classification based in a score that the higher is the value, the higher chance to have a baby, the higher chance to have aneuploid embryos, and also very nice link with morphology performed by the embryologist manually. So it's fully automating the process of a morphology evaluation, combining with morphokinetics is giving you a score which is linked with the outcome. Also, we perform a comparison between time-lapse incubator and standard incubator. This comparison was done in our clinic in Valencia, taking a look to the time-lapse incubators that we're having in our clinic. We have the Embryoscope, the Embryoscope Plus, the Jerry and Jerry Plus, and also we have these benchtop incubators, in our case, Aztec benchtop incubators. So we were using the benchtop incubator like a reference. The study was done in the clinic in Valencia. It was including a lot of cycles, close to 9,000. It was from 2 consecutive years, the last 2 years. The number of embryos cultured was more than 25,000. And the number of embryos transferred was 9,000 transfer embryos with a good proportion of single embryo transfer. So the first thing that we did was just split our results in autologous cycles and oocyte donation. You know that in Spain we do a lot of oocyte donation cycles. And taking a look to the standard cycles, we got very similar pregnancy rate between the Embryoscope Plus and Jerry, and also very similar implantation rate. Slightly different, slightly higher in some of the cases, but not significant by far. We take a look to the same results in oocyte donation. For pregnancy rate, similar results in the 3 instruments, Embryoscope Plus and Jerry, and similar also implantation rate with the 3 instruments. But what was very important, and we really was impressed, is when we put together the information of the 3 time-lapse systems and we compare with the standard incubator. Taking a look to all the cycles that we have done in the last couple of years, just doing an overview of the outcome, we got around 5%, between 4 and 5% more implantation rate. In this case was 5.5 more implantation rate when we compare the results from the time-lapse monitoring systems, putting together Embryoscope Plus and Jerry in comparison with standard incubator. So in our hands, in our clinics, taking a look to all the data together, of course that we are doing a more detailed study, but putting all the data together, we have seen that time-lapse is giving us better results than standard incubators with all the brands that we have. But we have been unable to see difference in the outcome between the different brands that we have in there. So trying to summarize what I have shared with you today, first of all, we have seen that this technology is interesting in order to observe the dynamic changes in embryo morphology. And these dynamic changes provided by time-lapse are allowing us to do a time-dependent categorization of the embryos, do a precise definition of relevant events of embryo development that are useful for embryo selection, for example, balastomer diameter, the trophotoid cell cycle. We have been able to identify atypical phenotypes that may interfere our perception of classical morphology. This is why classical morphology only does not work enough. It works for sure, but should be combined. And also, we must pay special attention to the behavior of the blastocysts after warming, especially the re-expansion and the thickness of the zona pellucida. We have seen that time-lapse technology is introducing the automation in embryology because we are able to perform full automatic annotations on morphogenetics and morphology. We are able to introduce some scores that are based in automatic algorithm that works for embryo selection because are combining both morphology and morphogenetics, but successfully because they're able to identify blastocysts with lower aneuploidy risk and also with higher implantation potential. Finally, we have seen that time-lapse technology can be comparable with standard incubator. We can be compared those. And we have similar results between all the time-lapse monitoring systems that we have in our lab, similar results. But when we pull all of them and we compare with the standard incubator, we see that the performance of time-lapse is still superior than the standard incubators. And finally, I would like to thank the team of embryologists, PhD students that are working in my team for doing research on time-lapse and artificial intelligence, in particular Raquel del Gallego, Lucía Alegre, Lorena Borli, and María Ángeles Valera, and also the students that I have this year, like Nando Meseguer and Celia Garcia, that are helping me to perform all these studies. And that's all. Thank you very much for your attention.
Hello, everyone. My name is Angus. I'm the Medical AI Director at Harrison AI. We build AI solutions in medical field. Today, I would like to present my talk on the predictions of embryo competence by image analysis using artificial intelligence. And my disclosure is that this work is sponsored by Virtus, Virtual Life, and Harrison AI, and I'm a co-founders of Harrison AI. So what we have on the screen here is time-lapse video recording of human embryo going inside a test tube for 5 days, dividing from 1 cell to 2 cells to 4 cells, and eventually into beautiful blastocysts. And hopefully we'll get one lucky parent, a little boy, a little girl one day. The goal that we have is to, in embryo selection, is to choose the best possible embryo. And the reason for this is that by identifying the viability of each embryo, we can prioritize the embryo with the highest viability and transfer those embryo back first. Even if it is a freeze-all cycle, we can always use this score to rank the embryos that underwent cryopreservation and select the most viable embryo for transfer. And the name of the game here is shorter time to pregnancies, to get the parents pregnant as soon as possible. Because of the importance of embryo selections, over the years, we have had many great techniques in order to standardize and improve the consistencies of the embryo selection process. The most commonly used technique is the morphological grading using the Gardner score or various adaptations of these methods, where trained embryologists use a scale a visual scale of various features, typically look at the trophectoderm or the inner cell mass and various other cellular feature in order to grade a blastocyst image, which get acquired near the end of the incubation period. As an alphanumeric score get attributed to each embryo, and this is typically used to select the best embryo for transfer. So there was a couple of challenges with this approach. First of all, as you can see, the timing in which we take the pictures is very important. And this is typically opportunistic and is often not clear when is the best time to analyze these images. You could be a couple of hours early or late, and we'll miss critical events that could happen to the embryo. The second challenge is a lot of people find is that this grading process is an incredibly subjective one. An embryologist have high inter-reader or intra-reader variability, and this has been shown in various studies in performing this grading. And because of that, that could introduce some errors or some inconsistency in the way we select human embryo. But most importantly, the grading system, remember, was created using knowledge distillation from human experts who have observed various embryo developments and in order to come up with this grading scale. And therefore, the knowledge in which we can extract from visualizing these embryos is only limited to the knowledge of the human expert who created the grading scale to begin with. There's been various attempts in using AI to automate this embryo selection process, such as paper published here, the STOR algorithms that look at using artificial intelligence to look at the blastocyst image and assign a quality score, good, fair, or poor quality on this embryo, effectively perform automatic grading of each of the embryo. The fallback here is, of course, these only mimic the scoring system that was used by the original embryologists in grading those embryos. And therefore, this algorithm provides some consistency and automation in embryo grading, but do not provide benefits in better embryo selection as compared to the original scoring, because that's what the algorithm was trained on to begin with. With the advance of technology, we currently have access to time-lapse incubation, where an embryo is incubated for 5 days, and a video recording of this process is available for the embryologist. So this provides much more information for the radiologist to analyze and observe the development of embryos, as compared to only looking at a single image at the very last day. The challenge, of course, is we've gone the other way. There's too many images to look at, and people typically come up with morphokinetic algorithms where they put various checkpoints at different key developmental milestones, and using the timing information and key morphological events during the 5 days in order to predict the viability of the embryo. This typically involves human annotators having to go in and laboriously put this annotation along the time-lapse video, and this scoring, this measurement is then fed into a scoring algorithms. Most commonly used is the SCID score that look at these T2, T3, T4, and PN event in order to determine the clinical outcome. This is typically a statistical model that look at non-clinical pregnancy outcome of these embryo. Once again, the challenges of this approach is that this is extremely manual and is extremely involved, where embryologists need to spend a period of time to do this annotation, and these annotations is inexact science, and there's great variability on this annotation. But once again, there's this knowledge distillation step where a human expert has made a determination that these timing information happen to be important. So therefore, we can never quite exceed what the expert knew about the embryological development process here, and therefore never really quite beat the status quo. There's also AI solution out there available that do automatic scoring and automatic annotation, which effectively automate the annotation part of this process, automatically going in and putting in these morphokinetic annotation. But once again, we are limited by the underlying performance of the morphokinetic algorithms. So the work that I've done in this field is in collaboration with the embryologists and the clinicians at Virtus, as well as Vitrolife, is to create a scoring system that can do better than what we already have. And we sat down and wrote ourselves a bit of a wish list on what would make a great embryo selection tool using artificial intelligence. And we realized that the first and the foremost important thing is that we need to be predicting clinically relevant endpoint. So there's a variety of algorithms in the market at the moment that look at various different endpoints. So the STOR algorithm, for example, look at the morphology and the embryo quality based on the embryologist's judgment. There's some algorithm that look at clinical pregnancy. Ultimately, what we want to look at is the live birth. Does this embryo create a live birth? And the more we move from the left to the right-hand side of this spectrum, the more clinically relevant the prediction is, but it's also harder to predict because of the increase in uncertainty. So we settle as fetal heart as a happy medium, where we have a good indicator of the quality of the embryo, but do not have to deal with some of the variability with maternal factors. And hopefully that will give good signals for AI to train. The second important characteristic of this system is that we want it to be completely end-to-end, meaning that we want the system to look at nothing but the raw time-lapse videos, so the 1 and 0, the pixel value of this video as it grows, and predict directly the chance of a fetal heart pregnancy in an end-to-end fashion. And as you can see, this is a highly dynamic event. And by looking at these videos, we can capture much more fine-grained details comparing to look at a single 2D images. The final wish list is that we want this to be a system that utilizes all of the data that is available to us through time-lapse incubation. So it's just a bit of a recap. If you look at morphology, this is a 2D approach where we look at the width and the height of the image at defined point in time and really ask ourselves a question: does this embryo look good? Whereas with morphokinetic, it is actually a one-dimensional problem where the only thing that we're looking at is the timing information and the presence of certain key developmental milestone in order to make our judgment about the qualities of these embryo. Whereas what we really want to do is a three-dimensional problem called morphodynamic, where we ask ourselves, how does the embryo behave through time and how does that changes? And looking at the entire embryo sequence give us a much richer understanding of the quality of the embryo and how it's developed. So the original work was called Ivy. We collect 10,000 embryos across 8 labs from 4 different countries from 2014 to 2018. We obtained clinical pregnancy outcome and fetal heart pregnancy for all of these embryo. And the most important thing to note here is that we include every embryo, not just the embryo has been transferred. So even embryo that has been discarded was included in this training, and that is labeled as negative fetal heart. An embryo with positive fetal heart is labeled as such if there's a fetal heart pregnancy at 6 weeks on ultrasound observation. And the reason for this design choice is we do not want this tool to just be a scoring tool for embryo that has already been pre-screened by embryologists. We want this tool to be a complete solution that automates the embryo selection process from an end-to-end process, which means that it must be able to rule out abnormal-looking embryos, embryo that are arrested, or embryo that have poor morphology that would have otherwise been ruled out by the human, and therefore do not require a pre-selection by embryologists before the algorithm can use. What we want to aim for is that this algorithm would decide on all of the embryos whether or not it would create fetal heart pregnancy or not. The details of this work has been published in Human Reproduction, which you can read in this article here. Deep learning is a predictive tool for fetal heart pregnancy following time-lapse incubation and placental transfer. So the exciting thing about this work is the algorithms learn tabula rasa, which is Latin for from a clean slate, meaning the model has no prior knowledge or assumption built in about the embryo developments or the way that embryo develops in 5 days. In fact, it doesn't even know that this isn't human embryos. The only thing it perceives of the world is a series of images which comprise of 0 and 1 values, the pixel intensities. The way that the algorithm works is that the model, which is a mathematical model of the human brain, look at these pixels and initially have random initializations of the connection of the neurons within this network and predict a random prediction, for example, 15%. Let's just say for the sake of argument that this embryo do end up create a fetal heart pregnancy. It would then compare this 15% with a positive outcome, which is 100%, and calculate a loss of, for example, 85%. 85% represent how wrong the algorithm is at making that initial guess. And we can use this loss to change the neurons within the neural network, a process called backpropagation, in order to make the algorithms better and better at predicting the viability of each embryo. And we do this loop hundreds and thousands of times, and eventually this prediction get closer and closer to the desired outcome, which is 100% for yes and 0% for no. And that is how the algorithm was trained and slowly improve in its ability to predict the fetal heart outcome. Here's a nice graph showing the training progress. The x-axis is the day since the training started on our computer, and the y-axis is the performance of the system in predicting pregnancy outcome, measuring using the area under the curve. As you can see, the algorithm started out making random prediction, hence the AUC is 0.5. And after 1 day, we're already at 85% AUC, and after 5 days, we're reaching AUC of 93%, which is an astonishing performance. So what it means in plain English of what 0.93% AUC mean is that if you randomly select an embryo from the negative cohort, and randomly selecting another embryo from the positive cohort, this particular algorithm, Ivy, will correctly rank the positive embryo higher than the negative embryo 93% of the time. So this score reflects the ability to rank and how often the algorithm can correctly rank these algorithms, which is ultimately what we want. And here's the ROC curve of these algorithms. So, once we got this work published, we quickly turned our attention toward trying to improve the system. And the question that immediately comes to mind is how can we do any better than what we already have? And one of the big thing about machine learning algorithms and that you hear a lot of is that these algorithms scale with the size of the data set that was used to train them, meaning the more data, the better the performance of the system. So it is quite natural that the progressions of Ivy, which is now called the IDA score as applied by Vitrolife, is that the algorithms now is trained on much more data. So this is the data set that is used to train the next iteration of these algorithms, the IDA score. As you can see, this data set is 115,832 embryos that's been used for the training of this system. And instead of spreading across 8 clinics, we are now across 18 different clinics, which is involved in this particular data set. So it's one of the biggest, if not the biggest, data set of human embryo with known pregnancy outcome ever been assembled. Within this, and this is over 10 times more than the original data set to train Ivy. Of this data set, there's a total of 4,000 positive fetal heart pregnancy and 10,000 negative fetal heart pregnancy. And of this, 100,000 was discarded embryo for a mixture of reasons, including poor morphological quality, arrested embryos, or aneuploid embryo as well, which have been discarded due to PGT results. So the big challenge that we look to solve with this massive data set is what we call the hidden stratification problems. Of course, the algorithms work just like before, whereas predict the fetal heart directly from the raw time-lapse video. But remember, fetal heart pregnancy is not a singleton concept. There's various gotcha that come with this. So for example, in real world, different clinics have different methodology inside their practice, including the day of incubation, anywhere between 3 to 6 days. And if you look at different days, there's also fertilization methods, IVF or ICSI. And within each of these methods, there's different cohort of patients to worry about, for example, age group. And as you look at this subpopulation and subcohort, as you traverse down this tree of possibility, each clinic around the world is actually quite unique in their lab methodology as well, the cohort of patient they do treat. And the question is, does the system that's been developed on a specific data set generalized to all of the subpopulation? And can this system work well in individual clinics around the world? Because that's ultimately what we care about. Does this solution generalize well? And using such a big data set give us the advantages is that we can answer some of these questions, which is previously impossible to answer. For example, we are able to divide the data set in 4 different cohort age group and still have one of the biggest data set in the world for each of the cohort. So here you see the IDA score algorithms performance in AUC once again in different age group. And this second generation performed even better than the 93% AUC of the original algorithms in almost all different age group. And the exciting thing here to see is that there's very similar and slight differences in performance, but we are still above the 90% mark in all age group. You might wonder, why does the performance of the system decrease slightly as the cohort get older? And this is not because the viability of the embryo goes down. I would hypothesize that as the age become larger and larger, there's the ability for the algorithm to predict the viability of algorithms decrease. And the reason for this is perhaps because maternal factors start to play a larger and larger role. But once again, this is hypothesis and it's probably require much more investigation. The other analysis that we've done was on the fertilization methods. So this is IVF versus ICSI. Once again, you can see that there's minimal performance difference between the 2 fertilization methods. Another variable that affects the performance of the system is the time of incubation. Because because of this system looking at a time-lapse video rather than a single image, the duration of the video matters. So here you can see a trend that the predictions of the algorithms get more robust if you get it longer video, because once again, there's much more information for it to look at, and there's low performance comparing to if the incubation time is shorter because there's less information for the algorithm to analyze. But once again, the performance is still great, above 93%, above 90%. And lastly, we did analysis on the lab that was contributed to data. Basically, we trained the algorithms on all of the lab except for the lab that is being tested, and then subsequently test the algorithms on that specific lab. As you can see, there's a variation in AUC across different laboratories, but always above 90%. So some labs, the algorithm perform a lot better than some other labs by magnitude of 5%, and this is probably due to the variation in the cohort and the methodology. Once again, I just want to stress that the high performance in the prediction of the algorithms in each lab do not reflect the qualities of the embryos. It reflects the ability of the algorithms to discriminate positive and negative embryos, and the laboratory methods play a big role here, as well as the underlying cohort of patient. But the bottom line here is that we can see a consistent performance of over 90% AUC in all of the 12 labs that we've done this tested on. And that's all that I have for today. Thank you for your attention, and I look forward to having you being in other talks. And we have exciting work coming across, which is a randomized controlled trial being run at the Virtus clinic in Australia and Europe, which will show the improvement, which we're looking to demonstrate the improvement in time to pregnancies using a system such as IDA score to assist the embryologists in selecting embryo. Thank you for your attention, and see you next time. Bye-bye.
So welcome to session number 2, the IVF laboratory. Our first speaker is Peter Schlegel, who is the James Cole Professor and Chairman of Department of Urology and Professor of Reproductive Medicine at Weill Cornell Medical College. He is the immediate past president of the ASRM and has been recognized for his clinical and research activities in over 300 published manuscripts with numerous national and international awards. And he is supported by research awards from the NIH and is a former trustee of the American Board of Urology. Our second presenter is Dr. Charlie Borman, and he is the IVF Laboratory Director at Massachusetts General Hospital in Boston and an instructor of obstetrics and gynecology at Harvard Medical School. His research interests include investigating the effects of maternal and environmental factors on oocyte and embryo quality and developing non-invasive methodologies for assessing human embryo development competency. Our third presenter is Dr. Carmen Rubio, who is the head of research for embryonic chromosomal abnormalities at iGenomics, with focus on non-invasive approaches of embryo aneuploidy. She specialized in cytogenetic studies in human reproduction, partly in the University of Barcelona. She has published over 100 papers in peer-reviewed journals, book chapters, as well as numerous lectures and conferences worldwide. Our next presenter will then be Dr. Brent Barrett, who is the Chief Laboratory Director for Boston IVF and oversees Boston IVF labs in Massachusetts, Maine, New York, Indiana, and Arizona. Dr. Barrett is currently a clinical instructor at Harvard Medical School and has authored numerous papers and abstracts in peer-reviewed journals and spoken at many national and international conferences. And he consults as an expert in laboratory quality and embryology. Our final speaker for the session will be Associate Professor Cecilia Slobloom, with over 23 years of experience in the field of ART. She trained and was awarded her PhD in embryology at the University of Gothenburg in Sweden, and in collaboration with the University of Adelaide. In her role as scientific director at Westmead Fertility Centre, she directs the embryology team and heads the Early Embryo Development and Epigenetics Research Laboratory.
Hello, this is Carmen Rubio from iGenomics Spain and iGenomics Foundation in the University of Valencia. I'm going to talk today about the non-invasive preimplantation genetic testing for aneuploidy as the new era for aneuploidy testing. How to perform embryo aneuploidy testing? At the current moment, the most common strategy is the preimplantation genetic testing, analyzing the trophectoderm biopsy and then doing the next generation sequencing analysis to these samples. As you can see in this slide, with this technology, we can identify aneuploidies for the whole chromosome, segmental aneuploidies, and also different levels of mosaicism. In this graph, you can see a monosomy for chromosome 16. And what is coming and what is going to be the new era, we believe, in the aneuploidy testing of the embryo is the analysis of the embryo cell-free DNA that is secreted by the embryos in the spent blastocyst media in which they are growing in the latest stages of development. As you will see in the next slide, we can analyze the media by NGS, a modified protocol of next generation sequencing, and we can identify also whole chromosome aneuploidies and segmental aneuploidies. The basis of this technology is that embryo cell-free DNA is released during embryo development and mostly at the latest stages of development from day 4 to day 6. And with higher concentration of this cell-free DNA, as the number of cells at blastocyst stage increases. This cell-free DNA released in the media can be analyzed, as I mentioned, in a non-invasive way by testing just the media without touching the embryos with next-generation sequencing without the need of a trophectoderm biopsy. And in the next slide, I'm going to present the experience we have accumulated in this field in the last 5 years, and I'm going to describe the results of the 3 publications that we have so far. So our first publication was a proof of concept, and we published 2 years ago in Human Reproduction. And in this study, we wanted to see if the spent culture media that had been in touch with the embryos had higher concentrations of DNA than control media without any contact with embryos. And in this study, we really observed higher amount of DNA in samples before amplification and after amplification. So this was showing that there was a release of DNA material from the embryos and that we were able to identify with the technology we had in our hands. Also, we were doing different studies to see if the incidence of the amount of DNA was higher in embryos that were aneuploid compared to euploid. We observed that the average, the amount of DNA was several, no differences between euploid and aneuploid embryos. We don't think that the main mechanism is going to be apoptosis in aneuploid embryos. We think are different mechanisms involved in this release of cell-free DNA into the media. Also, we were testing if there were differences in the gender, but there were the same amount in female and male embryos as detected in trophectoderm biopsies. And in this study, the main objective was also to compare the results of the trophectoderm biopsy and the cell-free DNA in the media and to do a concordance study. In this study, we were collecting the media in the embryos, in contact with the embryos from the day 3 to the day 5 in culture. And this study was performed using Embryoscope, and then the volume of media that we collected was 20 microliters. So in this study, the results were disappointed because the concordance rate in the results of the embryos between the trophectoderm biopsy and the media the concordance rate was only 30%, and we observed cases with partial maternal contamination and full maternal contamination in which we only observe normal female results corresponding to the DNA from the cumulus cells surrounding the oocyte. And we had close to 9% of non-informative results. So with this data, we were considering that there is something there is a cell-free DNA in the sperm culture media, but this DNA would not be suitable for a clinical test because concordance rate was low and the maternal contamination was too high. But we kept on working in this protocol and try to solve the problems we observed in our first study. So we started to do modifications first in the IVF lab part, and later on you will see in the molecular lab in the next generation sequencing protocol. So starting by the lab part, we did this pilot study in collaboration with Genera Labs from November 2016 to March 2018. We published this study this year in fertility and sterility, and this was really a proof of concept. In total, we were able to compare the results of the trophectoderm biopsies of 115 blastocysts and the corresponding media. And in this case, we did not use Embryoscope. We were asking the IVF laboratory to use the regular incubators, benchtop incubators. Also, very importantly, in this study, we didn't manipulate the embryos at all before the trophectoderm biopsy. So the hatching of the zona was performed right before doing the biopsy, but the media was previously collected. So the point is that we started to grow the embryos in the regular conditions in the IVF lab from day 1 to day 4. We asked to decumulate properly the oocytes to try to avoid any cumulus cells in the next parts or next steps of the culture. And then on day 4, we did a media change. And before this media change, we did extra washing. So we wanted really to clean again the embryos to discard the carryover of any potential DNA from previous stages, either from the embryo or from the cumulus cells, or even the potential contribution of DNA from the polar body. So at this stage, we clean it thoroughly the embryos, and then we transfer to a new drop. And in this study, the drop had just half of the volume, only 10 microliters of volume. And in this point, we grow the embryos from day 4 to day 5 or 6 at the moment in which the biopsy was performed according to the regular protocol of the labs. So we had the possibility of comparing the media with day 5 trophectoderm biopsy, day 6 trophectoderm biopsies, and just very few cases with day 7 trophectoderm biopsies. And from the molecular lab part, we modify the protocol for whole genome amplification to increase the input of DNA. So in this electrophoresis gel picture, you can see the results with the conventional ReproSeq protocol that we use for trophectoden biopsies and the modified protocol that we have incorporated for the media. For the media with this protocol, you can see very clear band, very bright band in all the cases, showing that we have really improved the DNA concentration with this protocol. So these were the two main improvements that we did, and these are the results. So we compare the results on days when the biopsy and the media was collected on day 5 and in the cases with day 6 or day 7. You can see how the informativity was 81.8% when the samples were collected on day 5, and the informativity was much higher when the samples were collected on day 6. Because as I mentioned in the first slides, when with higher number of blastocyst cells, we think that we are going to obtain higher number or high concentration of DNA. So you can see that informativity was much better on day 6, and if we look at the concordance rate between the trophectoden biopsies and the media, it was 63% when we were collecting the media on day 5 and 84% on day 6. So 63 is better than in our previous studies, double, but still not good enough. But 84% we consider is an interesting result. The concordance rate is much higher, and this really encourages us to pursue the idea of using the spent blastocyst media for clinical analysis of aneuploidies. In this study, if we just pay attention to the day 6 concordance rate, you can see how the false negative were 2.5 and the false positive rate was 0.5%. And we really need to improve these results. We are working and we have improved the protocol in the IVF lab to decrease contamination, because the false negative, we believe, are mostly because of the contamination with the cumulus cells, and now we learn how to get rid of these cumulus cells. And the false positive rate in most of the cases is related to degraded DNA, so we really need to work in storing and transporting the samples in the best conditions to avoid the degradation of this DNA. So this is something that can really be improved with a proper improved protocol. And one important finding that we had in this publication in fertility and sterility are the clinical results. So in this study, with these 115 embryos, the ones that were chromosomally normal after the trophectoderm biopsy were vitrified, and then they were candidates for transfer. And the transfer was performed according to this result in the trophectoderm biopsy. In the meantime, and in a blinded fashion, in the iGenomics labs, we were analyzing the media, but without giving these results to the IVF lab. And the IVF lab was performing the single embryo transfer when they were scheduled. At the end of the study, we matched the clinical outcome of the single embryo transfer and the results of the spent blastocyst media. And we established two groups, cases in which the media was also euploid, this 17 single embryo transfer, and cases in which the media resulted to be aneuploid, 12 single embryo transfer. And we were surprised by the fact that when the media was aneuploid, even though the biopsy was euploid, the miscarriage rate was pretty high, 50%. And this impacted really on the ongoing pregnancy and implantation rate that was only 16.7% when the media was aneuploid, and was as high as 52.9% when both the trophectoderm biopsy and the media were euploid. So this was suggesting us in this really small number of cases that maybe there is a decrease in miscarriage rate if we have also the euploid spent blastocyst media. So this was the pilot study, and at this time there were several studies published in this field. You have here a summary of these publications, and you see that the concordance rate between trophectoderm biopsies and the media ranged from 3.5% to 85.7%, the highest. In some studies, the comparisons were performed with polar body biopsy, as this one by Feitinger et al. And also in some studies, the concordance rate were calculated also with the full blastocyst. And in these 3 studies in which the concordance rate was compared also with the full blastocyst, the concordance rates were much higher. So again, pointing out the potential role of the media as a very informative source of genetic material. And this is one very important message from this slide. And the other important message is that in all these studies performed so far, the embryos were in some way manipulated before the media collection, because in most of the studies, even though the biopsy was performed at blastocyst state, assisted hatching for the biopsy was performed on day 3. So the embryos were in culture with this open window in which the embryos were releasing DNA. Also, in some of these studies, the embryos had an additional vitrification step, and the media was collected after thawing the embryos and culturing the thawed embryos. And we know that vitrification can have an impact in nuclear viability. So we think these are very important studies, that they reach interesting concordance rates, but in some way, the embryos had been previously manipulated. So we really wanted to have a protocol in which no manipulation was performed before the embryos are growing in the IVF lab, and the only manipulation is extra washes. That is not really, and media change, that is not really an invasive procedure. So based on this idea and what we have learned from our previous study, we developed this multi-center concordance study in which 8 centers all around the world were involved, and we aimed to analyze the results from more than 3,000 embryos, comparing the results of the trophectoden biopsy and the spent culture media. In this study, we are going to do the clinical follow-up until the end of the pregnancy, until delivery. And the idea is that we have 8 centers with different protocols, different media, different incubators. We wanted to see if we can have robust results in all the centers, and if this protocol can be expanding to several IVF centers. So in this study, we did the interim analysis. We closed the study in September, and we collected the data for the interim analysis in December of 2019. We analyzed the data and we submitted for publication, and now it's in press in the American Journal of Obstetrics and Gynecology. And the aim of this study was, on one side, just to analyze the concordance rate of the trophectoden biopsy with the media in this full amount of embryos, and in a subset of 81 aneuploid blastocysts, we wanted to compare the results of the media not only with the trophectoden biopsy, but also with the inner cell mass. Inner cell mass biopsy was performed in these aneuploid blastocysts that were donated for research after finishing the PGTA case. So here you have a summary of the results of this study. The average concordance rate was 78.2%, ranging from 72 to 86%. There were no significant differences among centers, and half of the centers had a concordance rate above 80%, and we feel comfortable now. We have the tips and tricks really to really suggest or to advise the IVF lab how to reach this above 80% concordance rate. And if in the sub-study, in which we also have the results of the inner cell mass, we were able to compare, on one hand, the media with the trophectoden biopsy, and for these 81 embryos, concordance rates were as high as 87.5%. The media compared with the inner cell mass biopsy had very similar results, 84.4%. So this is suggesting that the result is very reliable, not only with the trophectoden biopsy, but also with the inner cell mass, and is suggesting that maybe the DNA in this media is coming from both the inner cell mass and the trophectoden biopsy, because the results are very similar. Also, we compare the results of trophectoden biopsy with the inner cell mass that were 87.5%, so very similar to the results that we obtained with the media. The main source of discrepancies here of the trophectoderm biopsy with the inner cell mass were related to segmental aneuploidies and mosaicism, that we know that they have a mitotic origin mostly, but for full chromosome aneuploidies, the results were highly consistent and close to 95%. So these are the results from this study, accepted for publication, and we are still have many open questions related to this topic, and this is really a work in progress. We are doing basic research with Nicolas Placha lab because we want to understand the origin of DNA, working with live imaging of the nuclear material to see the dynamics of this chromatin during the latest stages of embryo development. We will need to understand what are the mechanisms in which the DNA is released. Maybe different replication occurs at the in the embryonic cells compared to somatic cells that can explain the release of these small fragments, and we will like to understand the mechanisms on how this DNA is released to the external compartment, to the media. Also, we wanted to investigate the clinical implications of the analysis of the media, and we have an ongoing randomized control study in which we would like to compare the implantation rates of patients doing regular single blastocyst transfer compared to patients in which the transfer is performed according to the results in the media. So the intervention group of this study includes patients in which the embryo transfer is performed according to the results in the media, and the control group is single blastocyst transfer in which the transfer is performed according to the morphology criteria of the IVF lab. The sample size of this study is going to be more than 1,000 single embryo transfers. We have so far 15 centers involved all around the world in all the different continents, and hopefully we will be able to collect the results of the ongoing pregnancies in the single embryo transfer and also the cumulative ongoing pregnancy rates. And we expect to have results in about 2 years. So this is the research we are doing so far, and we can conclude in the view of these results that the cell-free DNA from the embryo can be amplified and analyzed by NGS and looks like it's representative of embryo chromosomal content, that the concordance rates between trophectoderm biopsies and spent blastocyst media have been found as high as 84–85% when the media is collected on day 6, that there are high concordance rate with the inner cell mass, pretty similar to trophectoderm biopsies, and that this embryo cell-free DNA can be used maybe as a biomarker that give us important information of the embryo, and maybe without the need of a biopsy, we can prioritize which embryos have the higher chances of being chromosomally normal for transfer, and maybe with this, as we explained in our RCT, we can improve implantation rates. So this strategy will avoid embryo biopsy and would avoid the transfer of discarding embryos, because the idea is to use this as a prioritization system, not to discard embryos, just to select for transfers the ones with the highest possibility of being chromosomally normal. And finally, I would like to thank our mentor and the head of this research, Professor Carlos Simón, that is the head of the Scientific Advisory Board of iGenomics. I would like to thank my lovely team, Carmen, Lucia, and Luis, because we have been working hard in the lab to try to improve the protocols in collaboration with the IVF units. I don't want to forget all the IVF units we have been working with that really were enthusiastic about the protocol, and they were dedicated in improving the decrease in contamination. I would like to thank also the clinical studies team in iGenomics, Carlos, Emilio, and Marta, because they were establishing all the protocols to do the clinical studies and visiting the centers for the monitorization of the cases. And finally, to Nicolas Placha, a lab, for their contribution to the basic knowledge of these technologies. And also thank you to all of our colleagues in iGenomics all around the world, because they have given us the support and the advice to work with all the IVF centers we have been working with so far. And thanks a lot for your attention.
Hi, I'm Dr. Brent Barrett. I'm with Boston IVF. I'm the Chief Lab Director for 5 of the laboratories that Boston IVF has, and I'm here to talk to you today about IVF lab errors and how to prevent them. First of all, I have nothing to disclose, and just wanted to give you a brief outline of what we're going to be talking about during this session. Going to give a very brief overview of medical errors in general. Then the bulk of the talk is going to be a summary of a 12-year study which we did of errors in an IVF lab, in this particular lab in the Boston area. And then finally, we're going to spend a little bit of time at the end of the talk on some basic quality management principles and error prevention. So I want to start, as I said, to talk in the broadest sense about medical errors. In 1999, there was an Institute of Medicine report from the United States that was entitled "To Err is Human, Building a Safer Health System." The first sentence of that report stated, the title of this report encapsulates its purpose. Human beings in all lines of work make errors. And of course, this includes IVF. And it's very important that we, as clinicians, as embryologists, understand that errors are inevitable, they will happen, and that's why we need to talk about how to prevent them today. This was a paper that was published in 2016 in the British Medical Journal, and it estimates that medical error is the 3rd leading cause of death in the United States, or it was in 2013. And I'm showing this slide to indicate that this is not a small problem. This is a very large problem throughout medicine. And again, it's something that we need to focus on in IVF. A comment from the Institute of Medicine report was, as healthcare and the system that delivers it become more complex, the opportunities for errors abound. As those of you who have been involved in IVF are aware, particularly in the laboratory, but also in other parts of the facilities and clinics that we run, everything we do is becoming more and more complicated. From a basic point of view, we have very complicated procedures in the laboratory and in the clinical side of things. The documentation and records that we have are voluminous and are continuing to expand as we do more and more different types of things. The regulations that we're under, particularly here in the United States, but I'm sure in other countries as well, the College of American Pathologists, the FDA, CLIA, et cetera, we have to comply with all of those regulations. Increases in PGT, PGTA particularly, doing biopsies now on day 5, day 6, and in many places day 7. The increase in genetic testing. Many of us have time-lapse equipment and a lot of data to analyze with that time-lapse equipment. Frozen egg donor cycles. And then last but not least, certainly a tremendous increase in the number of frozen embryos as we do more and more cryo-wall cycles, the tanks, and keeping track of all of the embryos, sperm, and eggs that are in those tanks. In 2018, Dr. Sakis, myself, and Dr. Michael Alper published a paper in Human Reproduction where we looked at the types and frequency of what we called non-conformances or errors in an IVF lab. And that's what I want to talk a little bit about today and summarize. In March 2003, Boston IVF became certified to the ISO 9001, at that time 2000 quality standard. This is a standard that details the types of procedures that a laboratory or any facility, any manufacturing facility, as well as IVF, should meet in order to comply with the ISO 9001 standard. And one of the things that the standard requires is documentation of errors. And Boston IVF has created, and we currently maintain, an electronic database where we document the errors that take place within the clinic. This database is audited annually to ensure that all the records are complete. For the study, we looked at all the data from the laboratory that was entered into the database from March 2003 through December of 2015. We looked at all the errors that were in the nonconformance database during that period of time, particularly focusing on andrology and embryology. There were, during that time, over 28,000 IUI sperm preparations, over 25,000 semen analyses, and almost 37,000 fresh and frozen IVF cycles. Once we began looking at these data inside of this database, we realized that we needed to categorize all of these errors which took place. And so Dr. Sakis and I came up with the following categories that we divided things in. We'll talk a little bit more about what these categories mean in just a minute, but just a brief description. Communication refers to problems that occurred in between departments. So, for example, nurses to the lab or physicians to the laboratory, where there was a breakdown in communications, there would be an error. Documents and records, this applies to incorrect or incomplete documentation. Equipment and supplies, the failure of a piece of equipment or some type of supply that has a problem with it. External problems were things that occurred outside of Boston IVF, for example, in transporting frozen embryos or frozen sperm. Facilities would be a problem with the infrastructure of the building, such as the HVAC system. Human errors were an error that was most likely the direct result of a human action. And then finally, patient problems and complaints were anything that a patient initiated as a complaint. Next, once we had all these errors categorized, we also needed to grade them. Obviously, there are some errors that are much more significant than others, and so we put them into 4 different categories. The first category we entitled none or minimal. And this is an error, a problem, or could have been a near miss that occurred, but it was fully correctable or had absolutely no measurable effect on the patient's cycle. These types of errors often get documented, but oftentimes they don't as well because these are such small errors. So when we talk about these errors in a few minutes, we're not going to be talking about the full extent of these none or minimal errors. On the other hand, the moderate and significant errors, these are the ones we really want to capture. A moderate error is a serious error which affects the cycle, but where the entire cycle was not lost, whereas a significant error was termed a significant compromise or an entire loss of a cycle. Finally, and we're not going to be talking much about this during this session, but there are major errors. We don't have any of those here at Boston IVF, at least to date, but we needed to mention these. These are infrequent errors which have an extreme impact on a patient or a group of patients. And examples of these types of errors would be a pregnancy or birth that was confirmed to have arisen from a misidentification of gametes or embryos. Serious problems such as a tank failure that affects multiple patients, and systemic or repeated problems which affect multiple patients over a period of time. So for example, if a clinic or a lab didn't have a very good system for labeling straws, for example, or they repeatedly had documentation errors, that would be considered a major error. I just wanted to briefly give some examples of, these are real errors that occurred in our laboratory quite some time ago, but I just wanted to, as we moved through, as Dr. Sakis and I moved through these errors, and we decided on categorizing them, these are the types of things that we ran into. So this was an error, for example, that we categorized as a human error, and the severity was none or minimal. And in this situation, we had a culture dish, which was properly labeled, but the transfer dish, which the embryos were going to be transferred to, was mislabeled with an incorrect first name. The dish had the proper number of embryos in it at the proper stages of development and the correct last name and date of birth. So in a situation like that, after proper looking at this situation, we determined that it was not a problem. We corrected the dish and moved ahead. So there was no impact on the cycle. This, on the other hand, was a human error that had moderate severity, where we had a new embryologist who was handling dishes during a fertilization assessment. When he moved a number of the oocytes from one dish to the other, he accidentally flipped over the first dish, which had a few remaining embryos in it. 4 of those, oocytes, and 4 of those oocytes were not recovered. So this was a moderate error because it had an effect on the cycle, but there were 8 fertilized eggs which were not affected by this particular incident. Lastly, we have a significant error which involved equipment, and this happened to an embryologist as she was stripping some eggs and moving them. She had a stripper, and as she expelled the medium into the culture drop with the eggs in the stripper, no eggs emerged from that stripper tip. The pipette was examined under the microscope, and there were no eggs that could be seen in it, but there was a particle of plastic which was lodged in the tip. So the pipette was flushed and no eggs were found, but when the dish was re-examined, there were 7 ruptured zonas to be seen. So in this particular case, unfortunately, all the patient's eggs were destroyed, and this is obviously considered a significant error. So let's get to looking at a little bit of the data. So all of the errors that we found during this time, there were 360 of them, and of those, the vast majority of them were none or minimal, 277, with 66 moderate errors and 17 significant errors. And as we discussed before, there were no major errors. This is a pie chart that shows the breakdown of where in the laboratory these errors occurred. Not surprisingly, a little bit over half of them occurred in the embryology lab, approximately a third of them in the cryo or storage area. During this time period, of course, PGS, PGTA was not as common, so there were fewer errors there, just 5% in the andrology lab and then 1% outside of the lab in the transport area. This chart shows where the different types of errors occurred. So for example, you can see the first column there, and both of the charts refers to communication errors. The top chart are the errors that were considered none or minimal. The bottom chart, the blue, shows errors that were considered moderate, and red errors which were considered significant. So for example, in the first column, communication, you can see that there were quite a number of over 60 errors, almost 70, that occurred that were none or minimal in regard to communication. There were very few that occurred that were moderate or significant. So a lot of the communication errors turned out to actually be none or minimal. This was similar for documents and records, not quite as many, but again, no significant errors in regard to documents and records, but few moderate errors, but the vast majority were none or minimal. Equipment and supplies, we saw both none or minimal errors as well as moderate and significant. External, we'll talk about that in a few minutes. There were a couple of significant errors and moderate errors. And then the vast majority of moderate and significant errors, as well as none and minimal, came from humans. And this is not surprising in some ways, but it does point to the fact that it's critical how we train and how we prepare our embryologists to do the tasks that they need to do, because this is where the majority of errors are coming from. As we went through this exercise of looking at our errors and publishing this paper, we tried to find other publications that related to errors in IVF, as well as in the broader laboratory field. And we came across a few. There were a number of papers that talked about errors in a clinical lab, and you can see on the left there the percent of errors. And in a clinical lab, they can be as high as almost 0.5%. In transfusion area, 0.3% errors in that area. These are not errors, by the way, of transfusion where a patient died. These were mostly documentation errors for both clinical lab errors and transfusion errors. We did look at, the next column there is the Boston IVF endocrinology lab. We did look at the errors there. We're not going to talk any more about that today, but just to show that the endocrinology lab here at Boston IVF has a very, very low number of errors. If you look at the andrology and embryology lab, all moderate and significant errors are a little bit over 0.2%. And for significant errors, it's about 0.05%. There was an abstract that was published a number of years ago looking at the IVF lab mismatch rate that showed an error rate of approximately 0.1%, so a little bit higher than our significant rate. Complications directly related to IVF were very low, as well as complications related to IVF pregnancy were also very low, below 0.05%. What was really most helpful to us, though, in looking at the data and trying to compare our data with the data of other clinics was the HFEA data, which is published on a yearly basis. And we took the data from 2011 through 2015, and we show that in a chart here. The HFEA data labels things as A, B, and C. A is significant or severe. A is severe, which would be equivalent to our major errors. B is significant, which is the same as our significant errors. C is moderate, again, comparable to our moderate errors. And you can see the fourth column there shows the HFEA moderate and significant errors, B and C errors. HFEA has recently started looking at just errors that occur in the laboratory, and that's the light blue column that you can see that is about 0.15%. This is the laboratory alone, whereas the HFEA other numbers comprise both clinical and laboratory. And this compares, you can see the light blue line over on the far right there. This compares well with the Boston IVF moderate and significant errors, which came in at a little bit over 0.2%. So we feel that Boston IVF and the paper that we're publishing here is very similar to the HFEA data that we're seeing of an error rate, significant or moderate error rate of about 0.2%. So to summarize this portion of the talk, we had 99.8% of our cycles which proceeded without an error which affected the cycle, which 0.2% of cycles did have a moderate or significant error. Most moderate and significant errors were a result of human error, but also equipment and supplies and external factors were part of the problems. The error rate was consistent, as we just talked about, with HFEA data. I want to now spend a few minutes talking about quality management and error prevention. In that same Institute of Medicine report, they quote, "Most importantly, we must systematically design safety into processes of care. Errors can be prevented by designing systems that make it hard for people to do the wrong thing and easy for people to do the right thing. For example, when you get into your car and you have to push the brake pedal before you can take the gear shift out of park, that is something that car manufacturers have built in to make it hard to do the wrong thing. Similarly, in laboratories and in general in IVF and medicine, we should be trying to do the right thing and make it harder to make errors." I'm going to talk a little bit about some of these, but some of them we're not going to talk in any detail about due to the limitations of time, but I did want to mention a few of the elements of a good quality management system. Many IVF clinics do this, and that is they measure their quality through pregnancy rates, FERT rates, blast rates, etc. And I think that's in general a strength of the IVF field. One of the areas that we seem to struggle a little bit more is that one of the qualities of a good quality management system is that upper management is involved, and there should be a designated quality manager for the entire practice. This is a hard thing to do, but it's critical to have a robust, a good quality management system. Critical in IVF is controlling all the protocols, forms, and records that we have. It took us a long, long time when we did become certified to the ISO standard. We had over 3,000 documents that we had to control, and that took a long time, and that's something that's very, very important in IVF due to all the documentation that we have. Delivery of care. This should be documented with flow charts and process maps and should be carefully worked out, and we'll talk about that a little bit more in just a second. Personnel is absolutely critical in the laboratory, that an appropriate number of embryologists be there, that they have job descriptions, they're properly trained and properly supervised. And finally, we should have very good error documentation, corrective action, and improvement. And we'll talk about that again in another minute. So I wanted to talk first of all about how we design our processes, how we design the flow of work in our laboratories, in our clinics in general. I want to use an illustration here that's called the Swiss cheese model of accident causation. You can see in the left there in the green, it talks about the possibility for error. So you have a process or procedure, you're trying to prevent errors, and you can see the red line that goes through those slices of Swiss cheese down to an error. What you're trying to do is to misalign the holes in the Swiss cheese or not allow them to be lined up together in a way that would produce an error. And how do we do that? Well, one way of looking at it is to take each slice of cheese and to say, here's how we prevent errors in a particular way. So the first slice of cheese might represent the equipment that we have in the laboratory. Is it up to date? Is it functioning? The infrastructure, do we have enough space in the area? Is our HVAC system up to snuff as we need it to be? We also have to have protocols, documents, and workflow that is all arranged. We want things to flow smoothly and efficiently and not have a lot of chaos and confusion. Supervision and teamwork, another slice of the Swiss cheese. We want people to be working together under proper supervision where we have someone there who can help out when problems arise. And then finally, that we've hired the correct personnel, we've given them the right training, and that's really the last slice of Swiss cheese. All of this falls under a quality management system, where that quality management system is designed to misalign those holes in the Swiss cheese and not allow those errors to occur. Another way of using the same illustration is in a more specific way, and I've chosen witnessing here as the procedure that we look at in our laboratory. In witnessing, we want to have enough slices of Swiss cheese that we prevent an error by not allowing those holes to align, but we don't want so many slices of Swiss cheese that we're not able to do the work that we want in an efficient manner. So for example, we might use the first slice of Swiss cheese as the paperwork setup. Is that paperwork set up correctly? Are the names and IDs all correct? Are the dates of birth, the clinic IDs for the patients, are they all there as they need to be? The second slice of Swiss cheese would be labeling the dish. Another person labels that dish, and that dish should be labeled correctly with the patient's name, date of birth, clinic number, whatever ID you have on that dish. An initial identification by an embryologist of the name and the identifying information on that dish should happen. And then finally, some type of second human or electronic ID that should occur. These are 4 slices of Swiss cheese, which if used properly, will misalign those holes so that the possibility of an error does not occur. I mentioned before the number of embryologists and andrologists that are in the laboratory. This is a critical area, and not every laboratory has the same standard or needs the same number of individuals, but each laboratory should look very carefully at their lab and make sure that they have the right number of people, that they're not burning people out. This is a chart that shows the number of cycles, the ASRM minimum number of embryologists that they recommend, and then in a paper published in 2010, they showed what they feel was what the number of embryologists that high-performing labs have. You can see those numbers are different from each other, especially when you get to a higher number of cycles. And again, each lab needs to determine the correct number of embryologists for that particular lab. The nonconformances and completing these nonconformances and reporting these is an absolutely critical part of the quality assurance and the quality management system. When nonconformances are ignored, you lose out on a lot of information. On the other hand, when we report those nonconformances, we document them, we create responses to them, we look at them to see how we can fix them or remedy them, and ultimately we reduce these nonconformances. I remember when I first gave this talk a number of years ago to a number of people at an ASRM conference, and I had a laboratory director come up to me after I gave this talk, and he said to me, well, that was a great talk, but it doesn't apply to our lab because we don't make mistakes. And I just want to highlight that sometimes we can hide our head in the sand and not look at these nonconformances or errors as opportunities to make our practices, our labs better than they are. Finally, in summary, errors are inevitable. We need to acknowledge them and we need to use them to improve our clinical practices. We also obviously need to prevent them from happening as best we can by having a strong quality management system in place. We need to understand why they happen, get to the source of the problem, and ultimately that will allow us to improve the processes and have procedures in place for communicating and handling these errors. Finally, I would like to acknowledge all the embryologists in the Boston IVF Laboratory here in Waltham, Massachusetts, as well as Dr. Denny Sakis and Michael Alper for their help with the error study, and all of those who have been involved in the quality management program at Boston IVF. Thank you very much.